I think I'm gonna get started because it's 5 after. So if we all settle in. No, don't get everyone to move, it's fine. Anyways, so this is the GenPals Workshop 2021. Hopefully you're all in the right place. And welcome. This is really surreal having like an in-person event, because usually these kinds of things have been on Zoom for the past 18 months. And I will say I haven't presented in person since January 2020. So I'm just like, I don't know what's going on anymore. First, I just wanted to talk about like what is GenPals and why do we have this? So, you know, when we were all cooped up at home last year, every once in a while I'd have like a really insightful conversation with someone on Slack or via email with regards to something in the genomics world. I might have helped someone out, they might have helped me out. But it's really like conversations that would benefit more than just the two people engaging in them. And then, you know, I was conversing with my colleagues, Emmanuel, Fairly, and Axel, who are the other GenPals leaders, saying, you know, we really miss the kind of conversations you can have informally or formally with people kind of getting help on your research. So we started brainstorming about like, what could we do in the like pandemic Zoom world to kind of form a community of genomics researchers here at UCI. And we started bouncing around ideas, but what we landed on doing in winter and spring quarter of last year was an online seminar every other week where somebody presents a tool that they've used on their data. So it's kind of like a mix between a research talk and a journal club, where you can get help from an expert on how to use that tool. And then we thought that went successfully in the winter and spring quarter, but we realized that there was a certain barrier to entry to even understand what these talks were. The first talk I gave was on RNA velocity, which is a pretty complex and high level analysis. So it means that you have to already understand like what is going on in the entire single cell RNA-seq workflow to even get started on understanding RNA velocity. So this summer we were thinking, okay, how can we kind of resolve these problems? And what we thought was we could have a day where everyone learns the basics. And then that idea has been modified a little bit. Now we're stretching that out through the entire quarter. I think that's actually better because if you keep coming, you'll retain the knowledge over time instead of seeing it all in one day, and then you might not use it for a while. So if you keep coming back, you'll get a lot out of this, I promise. Okay, so the schedule for today looks like this. I'll give you this welcome and introduction overview to single cell genomics until 11:30. And then we're gonna have Liz talk about an experimental overview of single cell RNA-seq. Then we're gonna have some lunch. And then Fairly is gonna tell us about pre-processing sequencing data. So I kind of already told you about GenPals, but I'm gonna give you this visual metaphor of how I see it. So this burning building, this might, you know, stir some emotions, I don't know. But the way I see it is that this is your data, is the building that's on fire. And somehow you have to write your dissertation or write some paper based on this data. And your lab's already spent all this money on it, right? So you got to do something with it. And this is you, right? So you're walking into the burning building that's collapsing right before your eyes with nothing in your hands. There actually might be a better way to solve these problems than just doing nothing, which is bioinformatics methods. Although you might not know which method is the right one. You might not know who to talk to figure out what is the right method. So collectively, the group of firefighters is GenPals. You have people of different levels of expertise that can come together to solve the problem. I also like the guy who's holding the hose, but spraying totally away from the building. Anyways, so just, yeah, that's when Vivek tries to code. Not really, but, oh, well, this is recorded. Oof. So I just like got this, this is from a database called scRNA-seq tools, which is a website that tracks when tools come out. So if you can just see this, like kind of looks like, I don't know, it's not linear, but it's not exponential, but it's like going up increasingly over time. And there's over 1,000 tools that they track. Some are published, 70%. Most are not published, so they're not even like peer reviewed. But some of the non-peer reviewed methods are actually really good. It's really hard to figure out what are the good methods though, right? And there's all these different categories of methods. So there's like gene set enrichment analysis, there's data visualization, there's clustering, there's non-dimensional linear, blah, 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 right? So how the heck can I figure out how to analyze all this data by myself? And what we're trying to tell you is just keep coming back to our fall seminar and you will learn, right? And I'm gonna tell you exactly what the topics are that we're gonna cover. But first, I'm gonna tell you this little story that I put together of what it's like to do your first single cell genomics analysis. And I'm gonna kind of be portraying or like acting as if this were my first time. So this is kind of some of the things you would go through if it was your first time analyzing a single cell genomics data set. So, your PI is sitting there thinking about their favorite animal, their favorite organism, their favorite tissue that they're trying to study in the lab, right? They're thinking about where they're going to get their next pile of grant money. And, they've been hearing a lot about single cell genomics. They don't really know anything about it, but they've decided, okay, this is what we're going to do. They take you a lab meeting and they're like, okay, I have an idea. This is what you're going to do for your thesis. You're going to do single cell genomics. You're going to get all these papers. You're going to graduate, right? So now here you are alone after lab meeting. You're like, this sounds great. I'm sold. But like, where do I even start? Okay, so, first thing you got to do is you got to get your coffee. This is what you do in bioinformatics, right? You get your laptop. Then you start Googling for, okay, what are the tools I got to do, right? But, you find, okay, there's so many. What do I even do? And then some of these might have been floating around in your brain. You might have heard someone at some talk say like the word Seurat. You might have heard ScanPy. You might have heard 10x genomics. And so there's all these different routes you can go, but you're probably just gonna land on one of the things that sounds familiar to you, like Seurat or ScanPy. And together this kind of makes like the single cell genomics starter pack, right? So here you are, you've got your coffee, you've got your laptop, you've loaded up the basic tutorial on how to use Seurat. And you installed RStudio, you're all ready to go. And you don't actually, you're reading the tutorial and you don't understand exactly what's going on. But then you're like, I keep copying and pasting and I keep getting figures. Like all of a sudden I'm doing machine learning. And this was like, it's only been an hour. Like, this is actually like quite easy. I'm doing artificial intelligence, and you're dreaming about getting your Nobel Prize, which like probably won't happen, and then the way I see it is you're kind of at the top of the Dunning-Kruger curve where, your confidence is really high, but your skill level is actually quite low. So, okay, Sam, I can just copy and paste all this stuff from Seurat. Why would I listen to you? Why would I go to GenPal right? So many months pass, and after you've been practicing with the nice tutorial data, you actually get your first data set. So this is the data set I'm going to show you, which is from a publication from last year in Nature Medicine, where they did human and mouse single nucleus transcriptomics in Alzheimer's disease. So this is a data set I worked with in my lab, and we're just going to pretend this is our data for today. So as compared to the PBMC tutorial data set from Seurat, which only has like 3,000 cells, almost nothing wrong with it, very easy to analyze. Here we have 32 different individuals. We have different disease conditions. We have all these different cell types, right? Just to give you a kind of comparison of the two, these are coming from postmortem tissue that's been frozen. You have to actually do all the sequencing in multiple batches. The tissue might come from different brain banks. The samples might not all be the same quality. Different samples might have different brain regions. All these different problems are coming up that you didn't see in the tutorial. So you're like, okay, this is fine, right? I can just keep copying and pasting, right? So the first thing you do is you're just gonna okay, so what we're gonna do is reprocess this data set using the tutorial code, just copy and paste, copy and paste. The first thing we look at is just the quality of the samples. And when you got this plot from the ScanPy tutorial or the Seurat tutorial, there were only 3 of these violin plots. You're like, okay, I have all these samples now. The violins are different shapes, right? So I don't really know what that means. I'm just gonna keep going. Okay, so I do something called normalization. I guess I've heard of this before. I don't really know what that means. It's kind of a black box. But that sounds kind of fancy. It might be difficult. Turns out it's actually 2 lines of code. You can just not give it any arguments. There's no parameters. You just 2 lines of code, and it worked. Okay, cool. I'm going to take a quick side note to tell you about something called the curse of dimensionality. Tell you a little bit about it, but we'll get more into it throughout the quarter. But essentially, when you have high dimensional data, so we have like for each cell, we have 30 to 40,000 genes. It's actually just the more dimensions you have in your data, it's just harder to work with. That is called the curse of dimensionality. So one thing we do to resolve the curse of dimensionality is pick genes that are the most variable throughout the data set. And then we further reduce dimensionality doing a matrix transformation called principal component analysis. This is still, when you're doing the tutorial, this is still a black box. You don't understand what's going on. So, but you found out it's actually 2 lines of code. It's still really easy, right? Can't be that complicated. So you take, this data set had 33,000 genes. We just find the highly variable ones. We didn't even tell it like how many genes or any parameters, but it gave us something. So, and then, there's dots on the top that are different colors than the ones at the bottom. We must be doing something right? So now we're down to like 50 dimensions. Okay, that's more tractable than like 33,000. We can probably do something with those 50 dimensions. And just to kind of give you an idea of what this looks like, if we're looking at the first 2 dimensions of the principal component matrix, and these should contain the most variance in our data, I'm going to color them by different known marker genes of brain cell types. So neurons, microglia, astrocytes, oligodendrocytes, and so on. On the top left, we have a gene that's a neuron marker gene. We can kind of see that it's lighting up on one of these little tentacles, okay, and not the other one. So maybe we actually are separating our neurons from the other cell types here. And then right below it, you can see AQP4, which is an astrocyte marker gene. And similarly, you can see it's only in one of these little tentacles. Okay, cool. We might be doing something right. But if you've seen these single cell data sets, you might have seen these weird plots that kind of look like stargazing or clouds or something. And you're like, this doesn't look like that. This isn't right. So we have to do something called nonlinear dimensionality reduction. Now we're going from 50 dimensions and principal components down to just 2, right? So we're using UMAP here. Still really easy, 2 lines of code. And I'm only using 30 dimensions for principal components, right? Okay. So after this, you're like, finally, I can start looking at my data. So here's the UMAP, and I colored each dot, whether it came from an AD or control sample. So each of these dots are going to be one cell, and they are organized in this 2D space based on their similarity of their transcriptome. So broadly, we can see, okay, there's these like weird tentacles. This doesn't really look like what was in that publication, right? This doesn't really look like what was in the tutorial. Those were kind of more like these discrete units that represent the different cell types. Here, there's a lot of like mixing going on. I don't know why, okay, but like, look, there's difference between AD and control. Surely my PI will like that. And then you're like, okay, what are some other things I can color this by? Let's color by which sample each cell came from. And then you're like, oh no, why is it just like this gradient of the different samples? Shouldn't they just be organized based on what their cellular identity is? I don't know, all I did was copy and paste the tutorial. It must be right? And then let's look at the marker genes, right? So, okay, in the top left, we see, the neuron marker gene is lighting up in that top right region. But in that whole group, there's this like weird gradient where on the left side, there's no expression. On the right side, there's a lot. It's just like this nice gradient, but that looks really strange. Never seen anything like this before. And the last piece of like analysis we kind of see in single cell data is clustering. This is unsupervised machine learning. This is very fancy, right? This must be hard. This must take all day. It's 1 line of code, okay? So, and then we get this plot colored by clusters. And you're like, okay, I don't know what's going on. Like the clusters kind of should represent cell type or like a subtype, something biological. And then I'm showing you the plot from the publication on the right, right? So like, how do you even know what is the right number of clusters? How did these people settle on their number of clusters? And what do we do? Like, I didn't even give it a number. I just told it something, and that's what it gave me. It must be right? The other things we can start to look at are like, which batch was it sequenced in? And you can actually see that that's driving some of the differences in this UMAP as well. Something else is the total number of reads in each cell is influencing this. The percentage of reads that are mapping to the mitochondrial genome are influencing this. These are all things that you don't want in your data, but the tutorial didn't help us resolve it, okay? So now you're back at lab meeting and you're presenting this data to your PI and you show them this first and then they're starstruck. They're like, oh my gosh. Okay, so you're like, you're presenting and you're like, okay, I did all this stuff. I got these results, but I don't think it's like going that well, right? But they're just like, my grant's due, I gotta get my money. We gotta send this stuff out right now. But you don't really know what's wrong with your data, right? And all these thoughts start creeping in while you're going to sleep. You're like, is there ambient RNA contaminating my data? Are there doublets? Do I need to compare this to published data? Are there batch effects? Are there mitochondrial clusters? Do I need to use UMAP or t-SNE cross-species analysis? How many clusters do I need to do? I have a trait filter. When do I stop analyzing my data? Okay, so then you're like, I have to find someone who knows what they're doing, right? So if you come to the fall 2021 GenPals series, you'll learn in depth about all of these different topics, including pre-processing, how to use the HPC, because this kind of data won't run on your laptop, quality control, normalization, batch correction, dimensionality reduction, visualization, differential gene expression, and if we have time, we'll get into some more advanced topics. Anyways, that's the introduction and the overview. So please sign up for the Slack at this URL if you can take the time. The Slack is like a critical part of this where if you have a question, you can just like ask it and people will respond to you. That's the community part of this. The events are just one other part, but I think the most important thing, especially that the other organizers would agree with, is that this is a genomics community and somewhere you can get help. And also if you watch this and you're like, I know how to do all that stuff, why should I like be a part of this? Well, then you're the one that people will ask, right? So thank you for listening.